Morning, everyone. All
Good
right.
morning.
Sorry for being a minute late. Something funky was going on with some of the videos from last week that I was trying to fix. Okay, so let's get where we are going. All right, so where were we? All right, basically we ended up around here. Right, so this is a kind of basic, actually this is very much like the lac operon. So we've gone through all the kind of principles and ideas and concepts of transcriptional control in prokaryotes. And what we're going to do now is going to go through two examples. Those examples are the lac operon, which is involved in metabolizing lactose, and the trip operon, which is involved in generating tryptophan. Now, the lac operon is somewhat straightforward, depending on your perspective. And the trip operon is really tough, right? Students really struggle with that. And so before we get going, I want to share a general kind of idea with you all. Let me get rid of that. I'm going to bring it up the chat, make sure I'm not missing anything. Nope. And that's in bacteria, they are exceptionally good at turning on gene expression to very high levels, but then dialing things back as needed. And so from an evolutionary or kind of a population biology perspective, this makes a lot of sense because as soon as you discover a new metabolite as a bacterium, you want to colonize it as quickly as possible. You want to use it to generate energy and outcompete any of your neighbors that happen to be around and divide and divide and divide and divide, right? But on the flip side, it's a balance, right? If you put too much energy into colonizing that food source, for example, or that particular ecological niche, then you could spend more energy in kind of developing it than you gain in terms of dividing and increasing your population growth, right? Because all of this takes a lot of energy, right? So 3 units of energy, ATP or GTP, per amino acid, right? And these have lots of amino acids in them, right? So every time the lac operon is transcribed and translated, it costs a lot of energy. So bacteria essentially have two kind of modes when it comes to controlling gene expression for each operon. One is there's going to be a big, like, all right, flick that switch, and now we're going to the races kind of deal, right? In terms of turning on gene expression. And this can go from 0 to hero in like that. You know, it goes probably between 100 and 1,000-fold difference from a very low level, not 0, but almost 0, to 100 to 1,000 times the level of transcription. But they also have a dial. And it's funny, there's an advert in the UK with claymation with tortoises, you know, off and on again, right? So, and not going to make any sense to any of you, but it makes me laugh though. And so once you get up to that level, there's a dial, right, where you can dial things back, right? So basically, once you've started producing enough of what you need, now you can dial back the expression of that operon because you don't need as much of it, right? You still need some because if you don't have any, you don't use that food source or whatever it is, but you don't need as much. And so that's a way of kind of turning down the heat, right? Reducing the kind of like when you get some pasta boiling or whatever, right? You don't need it on like 10 the whole time. You just dial it back until it's kind of chuntering along at the level you want it at. And so bacteria have those two modes, right? And so each of these examples will talk about those. The main mode of turning on gene expression or relieving repression, hello doggo, of expression. And then the second mode, which is all about kind of fine tuning that level of expression. Okay. I'm sorry, I've got to let the dog out. She's licking my feet. I'll be back in a second. Sorry about that. She would just keep pestering me until I let her out otherwise. Okay, so both lac operon and the trip operon have that fine control, and it's quite different how it works. And actually, I think I can say this without reservation. It's the fine tuning that trips people up. The big on-off event for transcription in prokaryotes is pretty simple to get. It's really not that complicated. But the fine tuning, ooh, that's a real doozy or issue. There you go. Got to stop using that word because most people don't know what it means for some reason. Anyway, so when I'm going to kind of take you through the lac operon and what it looks like, basically. So where's a good picture of the lac operon? All right, so this is the lac operon here. Actually, that's a pretty crappy picture. It's too small. There you go. This is better. So as with that generic diagram above, there's two parts. There's the operon itself, which controls which has the promoter and operator, kitty cat, and the structural genes, right? These are the genes that do stuff. So the LAC-Z or Z is beta-galactosidase. This one turns lactose into galactose and glucose, right? So can't metabolize lactose directly. It can't go straight into cellular respiration. It has to be broken down into glucose and galactose first. I don't actually know what happens to galactose. One presumes it's used for something. I just don't know what. Probably modified and turned into glucose and then used as glucose. Right, so beta-galactosidase or LAC-Z is what does the work of turning lactose, this disaccharide, into two monosaccharides. And this glucose is going to be important, right? We're going to come back to that, right? There's a degree of self-regulation going on here. Shoot, which Greek letter is it? Oh yeah, there you go, Y. Greek letters, sorry, getting confused because the whole beta here. One of the other ones is a transport protein, and so this gets this disaccharide into the cell where it can be metabolized. And LAC-A is transacetylase, which no one really knows why it's there or what it does, but it's there. Presumably it does something important. Okay, so those three structural genes and the promoter and operator make up the lac operon, right? Don't forget as well, this operator, you can see it clearly here, it's between the promoter and the structural genes. So anything bound here will stop the transcription of these structural genes. Come on, love. The second part is a repressor gene, LAC-I. Now this doesn't in general have to be right next to the operon, but in this case it is. But it's a separate gene and it is produced constitutively, which means it's produced all the time, right? And so this encodes a repressor protein called LAC-I that binds to the operator and prevents transcription. Okay, so again, we've got three structural genes, promoter and operator, and those make up the operon and the LAC-I repressor gene. Okay, kind of give me a shout if you've got any questions. All pretty straightforward so far. And so essentially what we need to do if we're a bacterium is, if we're on a substrate or an environment where glucose is readily available, we'll use glucose because it's much more direct. Why bother going through all of this effort when you could just use this? So if glucose is present, we don't want to have a lot of this going on. If glucose and lactose, if glucose is present and lactose is not present, we don't want any of this, right? If lactose is present and no glucose is present, we want lots, right? So you can see there's a bunch of different kind of conditions here that we need to balance out the expression of the LAC operon. And so all of the control of the LAC operon is around meeting those or making the most of those three different conditions. And so the most clear condition is lactose present or not? If no lactose is present, there's no point doing all of this, right? It's just a whole bunch of energy wasted and resources used when they can't be put to use. There'll be no benefit to doing it. As I said to the other class, it's like if you studied for a psychology exam when you're not taking a psychology course, right? I mean, you could, but it's not going to do you any good, right? And it might actually do you some harm if you're not studying something else. Same kind of deal here. And so that's kind of big on-off switch. Is lactose there or not? Is controlled by LAC-I. And so LAC-I, in the absence of the inducer, right? So this is an inducible expression system. In the absence of LAC-I, will bind to the operator and prevent transcription. Now, in the presence of lactose, lactose now binds to the repressor protein, and it induces a conformational change in the shape of LAC-I. Now, that can happen before LAC-I binds to the operator. It can happen as LAC-I is bound to the operator. And in fact, in reality, what they're doing is they're constantly binding, releasing, binding, releasing. So it's kind of a bit of both anyway. But the net result is there's now no longer LAC-I bound to the operator. And so in the absence of that, now RNA polymerase can bind to the promoter, right? Because it's no longer blocked by LAC-I. And you can transcribe the LAC operon, translate it into the three different proteins that are required. And this is, go on, Tanya, I think.
Sorry. Yes, hi. So I'm getting lost. What doesn't bound to the promoter is the LAC-I?
So LAC-I can take two shapes. This is actually very common for particularly prokaryotic repressor proteins. So in one shape, they can bind DNA. In another shape, they cannot. And so really what you want to know is what causes those proteins to change shape and what's the consequence of that shape change and that binding. And so LAC-I alone has the conformation or shape where it can recognize the LAC operator and bind to it when there is no lactose present. Get you, there you go. So essentially it kind of goes between like different shapes. I think it kind of does that, right? It's a little bit odd seeing that. I probably shouldn't do that. But when lactose is absent, it's in the correct conformation and it will go in and it will grab that DNA and it will bind to it. in the absence of lactose.
And when there is lactose?
Then that shape changes.
And
so
it will still bind?
It will not bind. So it's kind of like that. So it can't like grab onto the DNA. It doesn't actually look like a pair of hands, obviously, but. And that can be through, it's almost like enzymatic inhibition, where you've learned how things can bind to enzymes and prevent them from binding the substrate or prevent them from having the correct shape, things like that. It's exactly the same. And I don't know if lactose does it by occluding the binding site of LAC-I, the DNA binding site, or changing its shape in such that it can't bind DNA. I'm not actually 100% sure which it is. But the outcome is that that protein cannot bind DNA if lactose is bound to it. So now you can see how lactose is the big switch, right? Because if lactose is not present, then that LAC-I protein will bind to DNA. It will bind to the LAC-O or LAC operator DNA more specifically. And if it's there, you don't get transcription. So it's a big on-off switch. That makes sense? Yeah. So yeah, with all of these and with the trip operon too, the repressors have two different conformations. I think LAC-I is it's lactose gets in the way. I think with the TRIP-R, the TRIP repressor, it's because it changes shape. But either way, it doesn't, in a way, it doesn't actually really matter because the outcome is that that protein will or will not bind DNA. Okay, so this is the big change. This is the big on-off switch. Now, as you can imagine, once you start producing a lot of this, which will be if you are on the presence of lactose, will be through the action of the products of the lac operon, right? You'll import lactose into the cell, you'll turn that lactose into galactose and glucose. Glucose levels are going to start going up. Now, this glucose is going to be metabolized, right, and turned into ATP, essentially. And so once the throughput of this pathway, turning lactose into glucose, starts exceeding the need or the demand for glucose, now you don't need as much of that going on. Does that make sense? So once you start producing enough of what the product is of this pathway, you don't need as much of that product to be produced. And so if you don't need as much, then if you just keep going gangbusters with the expression of that operon, you're actually producing more beta-galactosidase and permease than you actually need. And so you're wasting energy, right? And so now we need to, once glucose levels start rising, now we need to dial things back a bit. We're still on lactose, because if we're not on lactose, the whole thing gets shut down completely. We're still on lactose, but now we need to just kind of ease off on the throttle a little bit, right? Got up to cruising speed. Now we want to dial things back down a bit. And we're going to do that by sensing the levels of glucose. Okay, that's a key point. It's really important. And so, Go back to this one. We can't sense glucose directly. There's not a way of going, oh, we've got 350 molecules of glucose. We're kind of good. Our target's 300, right? Instead, what all organisms do, this is a very ancient thing because both bacteria and eukaryotes, we as animals, use this mechanism to sense glucose, right? And so essentially, our sensor is cyclic AMP. Right, and this is, I mean, this is the same for humans too. When your glucose levels drop, cyclic AMP goes up. And it's an inversely proportional sensor. So low glucose equals high CAMP, cyclic AMP. High glucose equals low. And everything in between as well, right? It's not a binary deal. It's a teeter-totter, right? Medium levels of glucose equals medium levels of cyclic AMP, for example. And so the lac operon needs a way to, or the bacterium controlling the lac operon needs a way of turning cyclic AMP levels into transcriptional output of the lac operon. So if you're producing, let's work through the logic of this, that may help. If you're producing lots of glucose, how much cyclic AMP will you have? If you got lots of glucose.
Low.
Low levels of cyclic AMP. Yeah, thank you, Pedro. You got that right too. How much activity of the lac operon do you need if you have high levels of glucose?
Low.
Cool. So low levels of, yeah, not as much is right as well, Pedro. Low levels of cyclic AMP lead to low levels of lac operon expression. Okay, so let's flip that around again. The opposite is also the case. If we have high glucose, if we have low glucose, right, we haven't turned enough lactose into galactose and glucose. How much cyclic AMP are we going to have? We're going to have lots. If we don't have much glucose, how much lac operon expression do we need? It's going to be lots. So high levels of cyclic AMP lead to high levels of transcription of the lac operon. Right, so exactly. I'll just add a little bit to that. You were right, Tanya. I just added a couple of little bits of information to make it even clearer. And so how do we interpret cyclic AMP levels, right? Well, and how do we turn cyclic AMP levels into transcriptional output? And so what we use, or what bacteria use, is another protein, come on, called catabolite activator protein, or CAP. And so CAP is a promoter of RNA polymerase binding. But only when it's bound to cyclic AMP. Right, so essentially we've got, it's almost like computer programming in a way. We've got a whole bunch of logic going on here. Glucose levels are low. Cyclic AMP is high. Cyclic AMP and CAP protein go together and increase lac operon expression. More lactose is turned into glucose. Glucose levels start going up. Cyclic AMP starts going down. There is less transcription of the lac operon, less glucose is produced, right? And so we have a kind of a fine tuning mechanism here, which responds directly to the output of lactose metabolism. And it does that by forming of this cyclic AMP catabolite activator protein complex, CAP-CAMP. So when there's lots of CAMP present, because there's not much glucose, that binds to CAP. That complex helps RNA polymerase bind to the promoter. And because there's more of it there, it's more likely to transcribe. And because we have lactose present, LAC-I is not bound to the operator, right? So basically, low glucose, presence of lactose equals gangbusters amounts of lac operon transcription.
Can the CAP work without the cap, or does it need like the cap?
It needs the CAP, the catabolite activator protein, to detect the levels, right? So really, cyclic AMP is a proxy for glucose levels, because and humans as well, right? Can't determine glucose levels directly, right? Or even ATP levels directly. What they do is they have to detect cyclic AMP, and they have to use other proteins. And I can't remember off the top of my head which ones do it in humans. But other proteins detect or sense that cyclic AMP level, and that gets translated into some outcome. And so the reverse is true as well. Once you've produced a whole load of glucose, you don't need full speed ahead on the lac operon. You can dial it back, and you do that by essentially you have very little cyclic AMP around. And so when there's not much cyclic AMP around, there's very little CAP-CAMP complex. There will be the odd one or two, right? But it'll be fairly rare. And so RNA polymerase will still bind, but not very well. And so that will result in a low level of transcription. And so essentially, you know, levels of glucose production will probably, start dropping. So cyclic AMP starts going up, and then CAP-CAMP starts recruiting RNA polymerase, and you start getting more lac operon expression, right? So it's almost like it's homeostasis, essentially, if you think about it. You know, it's like us and blood sugar levels, right? Blood sugar levels drop, release glucagon, blood levels go up, they overshoot a little bit, insulin's released, glucose gets absorbed, drops down a little bit. It's constantly kind of like bouncing around between a particular range of bounds. And it's the same idea with this. But this is at the level of gene expression, right? Okay, I'll give you a few moments to cogitate on that. See what doesn't make sense. All right, anybody got any questions? No. Okay, so again, remember, lac I binding or not is the big one. So if lac I is bound to the operator, it doesn't matter if no glucose is present, because, and it doesn't matter how much cyclic AMP is present. If lac I is bound here in the absence of lactose, you're not getting any transcription from the lac operon, or very, 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 very little. It's never absolutely zero. So whether or not lac I is bound here is the big switch. But in the presence of lactose, lac I will not bound. And now you have something between this extreme and this extreme. Right? So in the absence of lac I, now you can transcribe the lac operon. But how well you do so depends on how much glucose is present. If there's very little glucose present, right? So if you kind of put your hand over the screen and there's like nothing here, so I don't know. Hey, quit it. See if I can draw a little box. There you go. Right? You've got no glucose present. Okay, cyclic AMPs are going to be high. You want to have a lot of transcription from the lac operon. But once glucose levels start increasing, now you don't need to do this as much, right? Cyclic AMP levels are going to drop. There's going to be less cap-camp complex promoting RNA polymerase. Essentially, RNA polymerase is not going to do as good of a job of binding and transcribing this operon until either glucose levels go back down again because they're being used, or you no longer need to metabolize lactose. So that's the fine control. Okay, and I really want you to get these concepts because it's very hard to understand what's going on otherwise. It's just a bunch of words. Okay, cool. Now the trip operon is much more complicated. Sorry about that. So that's why we start with the lac operon first, because the lac operon, even with all of this complexity, is a little more straightforward. And so the trip operon, basically we are producing tryptophan, right? So where will you go, back? Leave the cats alone, dog. Essentially, this is, you know, a fairly reasonable facsimile of the trip operon, right? So the trip operon contains 5 different structural genes, all involved in the metabolism and generation of tryptophan. So there are 5 enzymes, but now we have a few extra wrinkles. So basically now we have a situation where the trip operon is on unless the product that it produces is present, in which case it is off. So if trip operon is normally on all the time, but it can be turned off, it's a repressible system. Lack operon is an inducible system. So the lack operon is normally off. And it's normally off because lactose isn't present. What are you doing producing beta-galactosidase? So the lack operon is inducible. The trip operon, you always need tryptophan, right? If you're going to be undergoing transcription. Sorry, Zoom catastrophe. I don't know what happened there.
I thought I was reading, like my internet went off or something. I was like, what's going on?
No, I think that was at my end for some reason. I don't know why. Probably just blame the kids. They might have jumped onto Zoom as well. Yeah, I don't know. I hope that recording didn't disappear. Fingers crossed, because that'd be a royal pain in the backside if it did. Yeah, it was funny because I was like, wow, Tanya just keeps writing the same notes in the same position for a really long time. And it was just because the whole thing had frozen and I was about to get kicked out. Anyway, so that's not as good of a readout as I thought. So anyway, you don't need lac operon transcription all the time, because if lactose isn't present, there's no point transcribing the lac operon. So the lac operon is inducible only when you need it. So normally it is off, but you can induce it to turn on. And the inducer is lactose. Now, if you think about the context of tryptophan production, oh, we've got a cat Zoom bombing me. There you go. Wow. She's probably looking at a squirrel on the street outside and thinking that it would make a tasty snack, would be my guess. Anyway, tryptophan you need all the time, because if you're in the process of translating mRNA into protein, almost certainly every so often you'll need a tryptophan amino acid, right? You need the building blocks to recharge those tRNAs so you can continue with translation. So the trip operon is always on. But once you have enough tryptophan, well, you want to turn it off, right? So the trip operon is repressible. So this is all about the trip operon being on, but you want to be able to turn it off once tryptophan levels reach a certain level, I guess. So, question make you think. If the trip operon is repressible and it produces tryptophan, what do you think will need to bind to the repressor to allow it to turn off the trip operon? So the trip operon produces tryptophan, essentially, or the products of it do. And it does so continuously. Once tryptophan levels increase to a certain point, you don't need to make more. Right? There's only so much tryptophan you need, right, at any one point. So you want to turn it off. So what thing do you think binds to the TRP repressor to turn off the TRP operon?
Is there something like the eye or no?
Well, there's a metabolite, but if you want to, okay, let's put it a different way. If you want to turn off expression of something that produces a metabolite, right? Once that metabolite gets to high enough levels, what do you need to turn off the expression? So once levels of tryptophan get high enough, what are you going to be detecting to determine if tryptophan levels are high enough? Tryptophan, right? So tryptophan is the co-repressor for the TRP operon. So remember, the TRP operon is repressible. So it's going to jump forward a little bit here. And so this is the opposite of LAC-I. So LAC-I will bind to the LAC operator in the absence of lactose, because it has to be induced to leave. Now we have the TRP operon, which has a separate regulator protein like LAC-I. This is called TRPR. Oh, sorry, Pedro, I didn't see your note in the chat box. Yes, you're 100% right. I'm sorry. Now, in the absence of tryptophan, you want TRP operon expression, right? Because you want tryptophan to be made. So in the absence of tryptophan, you don't want the TRPR repressor binding to the operator. So the TRPR repressor is an aporepressor. Right, and that something else is tryptophan. What are you doing, cat? And so that is its co-repressor. So only when tryptophan is present is that TRPR protein able to bind to the TRP operator. And that's the same as, I don't know what's going on here. I think my wife was calling me. Yeah. And this is the same deal as LAC-I in the absence of lactose. TRPR in the presence of tryptophan binds the operator and it prevents RNA polymerase from transcribing the operon. So protein bound equals no transcription. The big difference between the LAC operon and the TRP operon is in the LAC operon, protein will bind, LAC-I, in the absence of lactose. With the TRP operon, protein will bind only in the presence of tryptophan. Does that make sense? But either way, if there is protein there, I wonder who on earth that was. If there is protein there, you do not get transcription. Oh, some random ass person. Sorry, I was just checking to see if my wife had like broken down or something. Anyway. So this is, again, the big switch, right? In the absence of tryptophan, we have really high levels of expression of the trip operon. Soon as tryptophan level, oh, charismate, there you go, that's the substrate, I forgot that. As soon as tryptophan levels get high enough, and we're not talking about an intermediate level, we're talking about a high level, TRPR is then able to bind the operator with tryptophan and shuts down trip operon expression completely. So this is the big binary switch. It's either on, no TRPR, no tryptophan, or off, TRPR with tryptophan. One or the two. It's a binary deal. And so in some ways, it's a little bit simpler than the lac operon, which has a few more conditions that go with it. Right, so the lac operon is all about, is lactose present or absent, and is glucose present or absent? Right, so essentially you have a few different combinations. The trip operon is really just, do we have tryptophan or not? Right, and if we do have tryptophan, how much do we have? So in some ways, the trip operon is simpler in terms of the logic behind it. Some ways. Ha. Hopefully I've been preparing you for this, because this actually, the first time I taught it back in 2014, I was like, what the fuck is going on? Sorry, probably shouldn't swear in class, but I was like, I don't understand this, right? And so now I do understand it, but I appreciate that it is not easy to understand. Okay, so I am super happy to spend time and even drawing, if my dog will let me, the stuff out. Okay, so the trip operon has its fine tuning mechanism is called attenuation. I'm going to write some of this stuff up. And this is, I'm kind of hinting at it a whole bunch in the last section, this relies on the coupling of transcription and translation. And what is tryptophan used in as an amino acid? Question. Should be an easy one. What's tryptophan used for? It's an amino acid. Gets added to what kind of RNA? Begins with T and ends with transfer. Tryptophan is used in translation. There you go. So if transcription and translation are coupled, and tryptophan is needed for translation, now we have another means of detecting how much tryptophan is present, right? Because if tryptophan is not present at high enough levels, translation cannot occur. If tryptophan is present at high levels, translation occurs normally. So now we're going to use something that's going to be really potentially hard to get your heads around. We're actually going to produce a very small bit of mRNA that is going to be translated. It's not part of the structural genes. This is actually something called a leader sequence. So in a sense, we're going to produce a small, I don't know, dummy, and I don't mean that in a kind of derogatory sense, but as in a not used for something sense. Let me make sure I can see what I'm typing. Ooh, that was a good job. And that is going to be used to sense tryptophan levels. And when levels are high, I don't know, high-ish, that's not even really a word, but it will do. Tripoperon expression gets attenuated. And that basically means it happens less often. Now, I've got to be really clear here. The reason why I said trip levels are high-ish is that if trip levels are high enough, tryptophan is going to bind to the TRPR repressor and shut it down completely. Okay, so essentially we've got a space, right, if you think about it in this way, between no tryptophan and lots of tryptophan. I have no idea who that is. Hang on a sec. Oh, shoot. I expected you later, I'm sorry. Oh, I'm sorry, I'm teaching at the moment. I'm teaching. Give me just a second. See what the time is. You can come back in. Sorry, you can come back around 10:45. Would that be okay? Yeah. Yeah, just like that. I'll be done by then. No worries. Thanks. Oh, sorry about that. Someone coming to buy something at 3 o'clock showed up about 5 hours early. Okay, where was I? Right, so we've basically got this space, right, between no tryptophan and lots and lots of tryptophan. No tryptophan, we need a lot of tripoperon expression because we don't have any, right? Lots and lots of tryptophan, we don't need any tripoperon expression. That's when TRPR will shut down the whole lot. Okay. In between, we need to modulate how much tripoperon expression we have to maintain about the right levels of tryptophan. And that's what attenuation does. It kind of doesn't slow down transcription because you can't do that, but it basically reduces how often transcription is successful. right? So that sometimes transcription of the trip operon will occur and you'll get more tryptophan produced. Other times it will not be successful and the trip operon will not be transcribed. And as tryptophan levels increase, that rate of successful transcription decreases. So no tryptophan level, transcription all the time, every time. Tryptophan level starts going up, and then every so often, RNA polymerase doesn't work, turns off completely because TRIPR is now bound to the operator. So this is how we go from that binary on, off deal with the trip operon. We're basically going to start reducing how often RNA polymerase successfully transcribes the trip operon. Now in the lac operon, that's done by controlling how efficiently RNA polymerase binds to the promoter. Right, that's the cap-camp complex. The trip operon is very different. So the trip operon is going to use that dummy mRNA polypeptide to sense directly how many charged tRNAs have tryptophan on them. Right, and that dummy sequence actually has a proper name, obviously, because we're not allowed to call things dummies in science. That's kind of rude. Right, it's called the leader region. So just between the operator and the first structural gene, TRPE, is a region called the leader region. This leader region is transcribed. And this leader region encodes a polypeptide. That polypeptide does do nothing. It don't do no nothing. Maybe that's too many negatives. No. There you go. Doesn't do anything. It truly is a dummy polypeptide. Its sole purpose is to allow essentially the sensing of tryptophan levels. Okay. So hopefully I'm kind of like leading up to this, and this isn't going to be quite so like when we get to it. Transcription and translation are coupled. So essentially the translation of that leader region, right? That little mRNA or a little stretch of mRNA that doesn't really encode anything useful. Well, I mean, it does, but it's not going to turn into an enzyme, let's say, right? That little leader mRNA is going to get translated. And it's going to get translated as soon as RNA polymerase kind of moves along. You're going to have a ribosome following that mRNA as it gets produced, as the RNA polymerase moves along the DNA, because transcription and translation are coupled. Right? It's absolutely central to how this thing works. Can't stress that enough. So as soon as RNA, so there's no TRPR bound because tryptophan is not present at high levels. So no TRPR is bound to the TRP operator. RNA polymerase binds to the TRP promoter, and it starts transcribing this mRNA, right? It hasn't got into the structural genes yet, but it has produced an mRNA. Now that mRNA, as soon as enough of it clears the RNA polymerase, a ribosome will bind and assemble on it. And so you have literally RNA polymerase here going along the DNA and a ribosome right here going along the mRNA. RNA polymerase DNA, ribosome, mRNA. And so that little polypeptide that's being produced contains 2 trip operons. So what do you think will happen with that ribosome and the translation of that little that little mRNA if tryptophan is not present? What's going to happen to that ribosome? Is it going to be able to translate those trip operons if there's no tryptophan present? No, exactly. So if there's no tryptophan present, there's no tryptophan to add to trip tRNA. No trip tRNA, there's nothing that's going to go into the A site of the ribosome when it is over a trip operon. Okay? I'm trying to build up to this. I've still got, 12 minutes. We might not finish this today. Don't stress about it if we don't. I'm not. I want to really, because I really wanted to get this because it's actually pretty cool. I really think it's a super neat system. I do, honestly. You're laughing at me, Tanya, but it's true. This is very, if one were to design something, it would be a very impressive feat to actually come up with this design. It is very elegant. It really is. It's super cool. Okay, so
it's getting very complex. So I'm just trying to listen, like really listen, because it's getting complex.
It is complex. And so what I'm trying to do is I'm trying to kind of lead up to this process. Because if I just told you it straight out, I mean, I didn't understand it. And I was a postdoc at the time, or just had been a postdoc. Right. So it's not very intuitive. Right. And so that's why I'm trying to build up to this. So that you have everything kind of in there. And so when I tell you how it works, I'll be like, oh yeah, that kind of makes sense, sort of. Rather than I want to go home, my head hurts, or I want to go to some other part of my home. I don't know, right? Walk the dog or something like that. Anyway, okay, so transcription, translation are coupled. In that mRNA are 2 trip operons. So that ribosome will only continue translation if there is enough charged trip tRNA present. So only if there's lots of tryptophan around, again, not enough to cause TRPR to bind, but enough so that there's charged tRNA with tryptophans on them, will that ribosome proceed through that little bit of polypeptide. So that's why it's a dummy, right? That polypeptide doesn't do anything. As soon as it's produced, it gets broken down. The sole purpose of that polypeptide is to tell whether or not tryptophan is present. That is the sensor, right? So in the lac operon, catabolite activator protein was the sensor. In the trip operon, those 2 tryptophan codons in that dummy mRNA, those are the sensor.
Okay, so that's why it's called like the leader, because it goes first, and if there's enough, it's like, okay, go on, continue. If it doesn't, it's like stop. That's why it's the leader?
Well, kind of. This is where we get into the wrinkles. You've got the general sense. That leader is the thing that gets produced first, and it starts getting translated, right? It's not part of the TRPE structural gene. It is a very small, I don't actually know how long it is. I don't know, 100 or 200 base pairs long. it's very small. And the little polypeptide produced from it will also be very small. So you're right, if tryptophan levels are low, translation of that small polypeptide will not succeed. If tryptophan levels are high, it will succeed. Now, the consequences of that are the opposite of what you think. Okay? So if tryptophan levels are high, that polypeptide will be translated, but you don't want these genes transcribed, right? You want to dial things back. So if tryptophan levels are high, the leader sequence will be translated, but it results in the formation of what's called an attenuator sequence. That attenuator sequence kicks the RNA polymerase off the DNA. Now, there's a little bit of, in a sense, pointless revision for you. What's involved in intrinsic termination of transcription in prokaryotes? Not row dependent, although that's kind of in there too, but intrinsic dependent. What forms to kick RNA polymerase off of DNA?
A hair loop thingy.
Yeah, a hairpin loop. It's exactly what it is. Can you guess what the attenuator sequence forms?
Hair loop.
A
Maybe.
hairpin loop. That is exactly what it forms. And so if the leader sequence is translated, a hairpin loop forms, transcription is terminated. So essentially, this is a terminator sequence. This is like the sequence that you would expect to see down here after the TRPA structural gene, right? So down here, there's got to be something that stops transcription, right? So RNA polymerase just doesn't keep going off in the boonies. That may or may not, I don't actually know for sure, but that may also be one of those terminator loops. You also have a terminator loop in here, but it's conditional. It will only form if tryptophan levels are high. So if tryptophan levels are high, this leader sequence gets translated. That allows the formation of a terminator loop, a hairpin loop to form. RNA polymerase gets kicked off. Let me just type that out. Hang on, let's just type this out because it's a series of logic events, right? So the leader sequence then gets translated if TRP levels are high enough, and that's because there's enough tRNA TRP around for the two TRP codons in the leader sequence. And then, ta-da, a terminator loop forms and kicks RNA pol off the DNA. Equals no transcription of the structural genes. Ah, dang it. There you go, genes. And I think what we'll do is we'll go over this again on Tuesday at the beginning of class, because we can go through it in a different way, which I think may help. Also give time for stuff to sink in. And so essentially, I'll quit it. You've got the opposite can happen when tryptophan levels are low, right? If tryp levels are low, equals no or little tRNA trip equals translation of the leader sequence pauses, right? And prevents formation of the terminator loop. Okay. So this is how those intermediate levels of tryptophan are sensed. Essentially what the control of the bacterium is doing to control the trip operon and the kind of fine tuning is there enough charged tryptophan tRNA or tRNA trip around? If there is, that means the trip operon is doing its job, right? It's making tryptophan because there's enough tryptophan to get stuck onto tRNAs. If that happens, and you got 2 trip operons as well, not just one in that leader sequence. If that happens, that allows the formation of a terminator loop, kicks the RNA polymerase off, no transcription of the structural genes. Now tryptophan levels start going down, right? Because you're producing less of it because there are fewer enzymes around to make it, right? Because the trip operon expression is decreased. It's not off, but it's decreased. Now tryptophan levels start dropping, right? And they're dropping because they're continuously being used to make protein, right? They're amino acids. They're constantly being stuck into, these are your non-essential amino acids. This went back to a question from ages ago, right? Because bacteria can make all of their amino acids. And so they're making amino acids, they're using them to make proteins. Tryptophan levels start dropping. Now, these charged tryptophan tRNAs are being used to make other proteins, not just this one. Lots of other ones, all kinds of different proteins. So once the consumption of the tryptophan kind of increases and the tryptophan levels drop, it's not being replaced by the production of tryptophan because the trip operon is dialed back. Then you have fewer charged tRNAs around. Those charged tryptophan tRNAs are not going to be included into that leader amine polypeptide sequence. The ribosome pauses, right, and allows the RNA polymerase to get a little bit ahead. Terminator loop does not form. RNA polymerase transcribes the structural genes. So you see, I wasn't lying when I said that this was complicated. Okay, so I'll let that sink in over the weekend, or maybe you'll just go, I never want to think of this ever again, right? That's quite possible too. But at least you got to think of it for a little while, right? Until we get through exam 4. Have a think about that. Look through the rest of the slides. So I've taught this in a different way to what it is in the PowerPoint. And Monday, we'll go back to this and we'll finish off these few slides. And essentially, there's some real nice pictures which will show how this happens. Okay, so have a think through all of that. Have a look through this. And always keep in mind, what is the purpose of this control? Right? What do we need from the expression of this particular operon? How do we determine once we have enough of that? Right? How do we determine if we have the substrate present at all in the case of the lac operon? Right? Think about what's the purpose of this operon, because the purpose of it in terms of what it produces will inform how it is controlled, right? And that will help a great deal in understanding the mechanism of how it is controlled. All right. Otherwise, honestly and truly, it's just a giant mess of words. And it'll be very hard for you to understand and be successful when I go to great lengths to test how well you understand this in the exam. Yeah, I'm sorry. I wish I could be that nice professor. No, don't worry about it. We don't have exams, but we do. But anyway, okay, I think that's enough torment for now. I shall see you on Tuesday. Really, truly do go through this, figure out what you don't understand. Do not expect to understand all of this immediately because you won't. I've been teaching this for years and I do understand it, but that's because I've done it so many times, right? Don't expect to understand this immediately straight off. Go back through it, read up about it, watch videos, go through the PowerPoint, make a list of questions to bring on Tuesday, and we'll spend a little bit of time doing that on Tuesday. Okay? Cool. All right. Have a great weekend, everybody. See you on Tuesday.